Correct.
Okay. Okay. Now let's look at the reproduction and growth of bacteria. Okay. Normally when you say growth, people are thinking about things increasing in size, becoming bigger. In bacteria, when we say growth, we imply increase. This means increase in the number of cells, not the size of cells. Now, as to how fast a bacterium will grow, the growth rate, it will depend on the type of bacterium. Two, it will depend on environmental conditions. When we talk of environmental conditions, we're talking about nutrition, temperature, and pH in the environment the bacteria finds itself. Please recall the things that you learned in year 1 about the curves, the bacterial growth curve. Remember, there was the lag phase, the whatever. We will use those when we talk about sexual reproduction. For now, let's talk about asexual reproduction. Asexual reproduction simply means that we are going to have an increase in the number of bacterial cells without involving another cell. Okay, let's quickly go through these and see how the bacterium accomplishes this. The most common method is transverse binary fission. I've used the abbreviation here so when I ask you what it is, don't just go and write TBF, write transverse binary fission. Okay. When you describe something, please don't use the terms in the question. Okay. I will explain it to you shortly in the next one. Okay. Now, how does this happen? We have a single cell which develops first it will enlarge, and then it will develop a transverse septum, or a partition that is perpendicular. Then it will divide into 2 organisms. I've reminded you that this division is made possible by the intervention of the mesosome. Now, budding. If you're asked to describe budding, if you say the cell produces a bud, you don't get anything, because the person is asking you about budding because they don't know what a bud is. So you have to say a cell produces a small protuberance or a small outgrowth, and this outgrowth is the bud, and it develops on one end of the cell. Now the bud will enlarge and it will develop into a new cell, and after developing into the new cell, the cells will separate. It's a pity that you know what budding is, but when somebody says describe it, you say bud is produced and then 2 cells. You don't even say the cells will enlarge, the cells will separate. Please just treat me like I'm an idiot, because I am. Okay. Sometimes during budding, the cell will produce an arm first, and this arm is called a prostheca. And then the protuberance will be born at the end of this prostheca. And the bud will then enlarge, and then the 2 cells will separate at the prostheca. An example of a cell that will do that is Hyphomicrobium vulgare. Please, in order to know these things, just pick up. I've given you lots of names of bacteria. To get that extra mark When you're writing these notes, which I hope you will be doing, when you write, add an example. For each one, add an example. Fragmentation is an asexual process which is normally produced by filamentous, or the long rods which I told you about. The long rods will break down into smaller pieces, and these pieces may be rods, bacilli, or they can be cocci. And each bacillus or each coccus will then elongate to produce another filament. An example is nocardia. Exospore formation. Exospore formation is achieved by the formation of these partitions or septa at one end of the hyphae or of the filament. And each partition or a spore gives rise to an individual, and the individual will grow into whatever that bacterium is. An example here is streptomyces. Now, endospore formation. Endospore formation is not really an asexual process, but I've put it here because it is the way by which the fungus will survive during difficult conditions, inconducive conditions. Now, let's say, for example, it becomes too dry, or there is no food, whatever, and the fungus doesn't want to, the bacterium doesn't want to die. What it does is it secretes a wall around a particular part of itself, and that part should have nucleic acid in it. And that hard spore is going to last for a very long time. If, for example, it was dry conditions, until wet conditions come back, and we hope those wet conditions will come back with food, and the bacillus will, the bacterium will then get out of the spore, germinate, and start reproducing more members of itself, but by any of the asexual processes we have outlined earlier. Please note, not all bacteria produce endospores. There are certain bacteria that produce endospores, and sometimes you use this endospore production to identify the bacteria. Here I've given you two examples, Bacillus and Clostridium. These two will produce endospores. How is this endospore produced? I've already said it very briefly, but I've outlined it here for you. On the outside, we have the cell wall, and in red, it is the cell membrane. There will be invagination of the membrane, but before, as that mesosome goes in, the circular nucleic acid separates by the aid of this mesosome into two equal parts. This part is going to be lost, and this part is the one that is going to be saved. Because this part is going to be saved, this part generates a wall around this particular part. So you see, here we have the spore, and this one is busy surrounding it with more and more wall material, to a point where we have the nucleic acid here with its own wall, and the other double wall that was made by this one. So here we have a hard wall. This one will die, This will shrivel off and die off, but this will stay. So here we have an endospore. The steps that are used, that are followed, are outlined here. This is called a four-spore. There will be a slide later on that will show you the layers. We have an exosporium, etc., etc., which are the layers that cover the endospore. Virtually, that is asexual reproduction. Sexual reproduction, in brief, sexual reproduction is change in the DNA sequence of a bacterium such that, so when we're doing this sexual reproduction, please don't care about changes in number. Here, we are going to look for change in the sequence of the DNA of the bacterium at the end of the process. Once there is change in the DNA sequence, then we have sexual reproduction. When your father contributes and your mother contributes in sexual reproduction, the stages that new nucleic acid goes through produces a new combination, which is you. Please note, even if there are 100 of you by the same father and mother, you will never be the same, because during that sexual process, there is something we call chiasma formation. That means the sequence of the genome of your mother and that of your father goes through a change before it contributes towards your formation. In the formation of the gamete, in the formation of the egg, there is meiosis, and during meiosis, there's chiasma formation, which brings about a change in the sequence of the bases in the chromosome. Your father does the same, your mother does the same. So the four products of meiosis are never the same. And we don't know which product from your mother will combine with which product from your father. Now remember, this bacterium has 1,000 micrometers of nucleic acid. It is going to bend and twist and bend and twist and separate, and the combinations will be a lot. So please note, we are looking for change in DNA sequence. So remember your DNA, and let's look at how it changes. There are basically three methods in which the sequence will change. One, it means, okay, in bacteria, we don't use male and female, father and mother. In bacteria, we use plus and minus, unlike poles will attract. The one that in your case you call your mother, the one that your mother receives from your father. So it is called a recipient. The other word for your mother would be a recipient. Your father would be called a donor. So here we're going to see that there is a recipient and there is a donor. And please note, the donor doesn't give us everything. It just gives us a fragment, a piece of its DNA. It doesn't give us its whole DNA. And that piece which it has given us is is treated as what? The guest of honor. It is accommodated by the recipient. And in the process of accommodation, it means the recipient has to throw away part of itself. It's just like saying if at home you have 6 seats at table and then a visitor comes. If the visitor is your friend, or you are of the same height and everything, then the visitor will sit on your chair and you will have no chair to sit and you'll be thrown out of the table. So as to where the visitor will sit, it depends on whose visitor it is. Okay, so keep this in mind as we go through the process that are going to follow. Thank you. All right. Oh,